Okay, I think I'm going to go ahead and just get started while people are logging in and want to make the best use of your time. I'm Jessica Long. I'm here at Whitney Laboratory and I work in advancement. And so we're bringing to you our December Sip in Science. We're excited. This is our fourth one. And it's just been really fun to take this journey with you guys through the different research and programs here at Whitney. We will be continuing it coming up in January and into the spring. We'll see how it goes. We miss having you all at our Loesgen Auditorium in for talks, and we hope to get that going when we get through this difficult time, but we're so glad you joined us online. We hope you and your family and friends are doing well and safe over this next holidays that are kicking off actually today and into the near future. So we hope you're doing well. Just a couple housekeeping things we want to let you know. January 14th is our next talk, and actually our director, Dr. Mark Martindale, will be joining us. He will be talking about marine labs and the interesting things that in general that we study, and really into some of his research on regeneration and some of the neat creatures that he works with. So I hope you can join us for that next one in January in the new year. As you're watching the webinar, if you haven't joined us before, we actually don't see you. You guys can see us, but the way that we can participate is by you logging in and asking questions. We have a poll that goes up as well. And I'm going to have Patrick Thompson here with Whitney. He's going to tell you a little bit more about the Q&A.
Good evening, everyone out there. So for the question and answer session, you can write your question down in the chat next to participants. It looks like a little talking bubble. Just go ahead and click that bubble, go ahead and enter in your questions, and then we'll try to ask them at the end of the presentation.
Thanks, Patrick. And just in your view, if you want to click up on the top right hand corner, you'll see speaker view on most Zooms. So that's probably the best way to see our speaker and what's happening here on the screen. So without further ado, I want to introduce to you our next speaker, Dr. Sandra Loesgen, who joined us in September from the Oregon State University. She graduated summa cum laude with her PhD in organic chemistry and pharmacology from the George August University in Göttingen, Germany. After that, she was a postdoctoral researcher at Scripps Institution of Oceanography and then at the National Institutes of Health. She then served as an assistant director at the Oregon State University, an assistant professor, and joined us in chemistry in September, as I mentioned. So we're really pleased to have her with us, and let's get started with her talk. Thank you.
Here we go. My apologies. Thank you so much, Jessica, for the nice introduction. Welcome you all. I'm glad you're here today and joining us at the Whitney Lab. I'm excited to talk today about research going on here at the Whitney Lab in my laboratory. And it's gonna be a little journey telling you stories about drugs and how we can find drugs in dirt and in the ocean. Before that, I wanted to give a little bit of background to get to know each other. I am German. If my accent doesn't give it away, it's definitely this slide. And as Jessica Long mentioned, I did my PhD in Germany. So here right in the center of Europe, my family lives there, and I studied chemistry and pharmacology there and got a doctorate degree. I wanted to share this fun picture of myself. We have this really fun tradition that all the doctorates that are leaving the University of Göttingen receive such a doctoral hat. Looks a little different than the doctoral hats we have in the US. And this is a tradition that is really common throughout Germany. And it's just a fun way of all the colleagues and all your peers to honor you and your work during this journey to receive your PhD. So this tradition of making a doctoral hat actually followed along to my laboratory. And here you see some of the recent hats that we made in the laboratory for doctorates that came out of my laboratory. So several students that graduated recently, and you can see how much passion goes into forming those doctoral hats, telling stories about the students and their journey. And just recently, in November, we were fortunate enough to graduate Paige Mondelaire. So a picture of her during this challenging times of COVID. Obviously, most of the seminar were given in Zoom. And as you can see, everybody is masks here. And we are really proud of our graduates from the Whitney Lab. And as you can see, the tradition lives on. A doctoral hat for Paige was made, honoring her time here and her research. So a little bit of background about my lab. I want to go on and talk a little bit about the drugs that we are trying to find new ways of treatments for various human diseases. And while we are thinking dirt and oceans are a good source to look for new chemical entities. So I want to start off and introduce to you and maybe take away the fear of chemistry. So natural products or natural product chemistry is everywhere. And as you might have used all before, aspirin is shown here on the right of the slide as a pain relief medication and a blood thinner that is derived from a tree. And that is, I think, also a question coming up. So plants have been the source for medication for centuries. Morphine, one of the opioids, enabled us to perform surgeries, obviously also having a potential to be addictive. Down here on the slide is an anti-tumor drug, Taxol, that's been isolated by the National Cancer Institute from the Pacific Northwest yew tree. And this is one of the most used cancer drugs currently administered in the US. On the far left bottom, you see artemisinin. This is an anti-malaria drug. And you might have heard the story how in Chinese traditional medicine, the plant was already used as an anti-parasite. And it took a team of researchers years to figure out in old scripts which of this plant, how to process the plant to make a good drug. So chemistry is everywhere. And also microbes, shown here is a kind of a mold on a nectarine or an orange. And so fungi and bacteria also producing chemicals. And here you see the antibiotic penicillin, as well as other compounds. So natural products chemistry is everywhere and has been used to inspire drugs. The poll is okay. Let's keep going. And so natural products have inspired our drugs that we're using on a daily basis. So the FDA is approving in the US drug entities, and 75% of all of these drugs that have been approved over the last 40 years, and even longer if we could study these times, have been come from nature. So nature provides us with the means to treat various diseases. And while I spent time on talking about the plants here shown in green, the fungi and bacteria also wonderful chemists. They produce structures like penicillin or the antibiotic streptomycin that are used in the clinic as antibiotics. So we are interested in the microbial chemistry and how we can use this to find new drugs. I want to give a little bit of background about these bacterial or microbial chemicals that we have been using as antibiotics for a long time. This is a slide showing two gentlemen studying what looks like agar and mold in vials. And most of you might have heard about the gentleman to the right, who's Alexander Fleming. And the story obviously goes that he studied bacteria, but by serendipity, he found that fungi on the same agar plates were inhibiting the growth of the bacterial neighbors. So this is a kickoff story for penicillin, a beautiful organic structure here that we're using in several generations now as our antibiotics. So 4th generation of penicillins are used in the clinic. And penicillin is truly a remarkable story. So Fleming observed that this mold can prevent his research looking into bacterial growth, but he didn't stop there. So he followed up on this observation and the story starts here. I have to mention that penicillin is not the first antibiotic. It's often considered one of the earliest, but the sulfonamides, these are dye molecules that I would consider the first antibiotics brought into the world. So these are small synthetic dyes that have been developed in the early 20s and 30s by Bayer in Germany, coming out of the dye industry. And these are really the idea of having a chemo drug, to have a golden bullet that will be taken orally and fight diseases, fights infections. At the time, gangrene and other bacterial infections, there were no help and no rescue. So this is the first drug in place. But going back to here's Protosil. Protosil is this dye. It's a dark red colored injectable medicine that helps against bacterial infections. Going back to Fleming, it took a while to test penicillin, and Florey and Shane, both at the University of Oxford in England, were testing penicillin once it was cultured. So we grew the fungus, it was cultured in large scale, and then introduced into mice. So this whole idea of testing a chemical entity into a living organism before we're giving it to humans, that was really developed around penicillin. All the testing we are currently doing for all kinds of vaccines and drugs are all relying on these early insights that animals are required for testing. During World War II, penicillin was mass produced in large quantities in the UK and in the US, and it was used in many ways, right? Gonorrhea, it heals in 4 hours. What a promise, right? So it changed our lifestyle. It changed how we treat bacterial infections. But I think one thing that we might overlook is it's not just the bacterial strep throat or the ear infection. Without antibiotics, there wouldn't be any kind of burn management. For severe burns, you will be put on antibiosis treatment. There wouldn't be any other open heart surgery or replacements of bones, etc. There were no transplantation of organs possible if we couldn't put a patient on antibiotic prophylaxis. And 70% of the population might not be here today if penicillin wouldn't have saved the day. I'm allergic to penicillin, many of us are, so luckily there's more than just one chemical entity that helps us. If you're interested in these early antibiotic stories, this is a book that I enjoy reading. Thomas Hager is the author, and this is focused on the early Prontosil and other drugs, how they've been developed. Very entertaining historic novel that I can highly recommend. So penicillin was a game changer. But on this slide, another gentleman, a contemporary to Fleming, were working with actinobacteria at the time. And so Salman Waxman, another Nobel Prize winning microbiologist, found that many of these actinomycete bacteria, lots of them found in the soils outside, are making drugs as well. So they're producing these macrolactam or macrolactone macrolide antibiotics. Some of them you see in the clinic shown here. Also streptomycin, the to-go drug for tuberculosis treatment, is coming from one of these bacteria. So bacteria are also very potent chemists, very prolific in producing structures that we can use for our infectious diseases treatments. So microbiomes is in everybody's ear, right? So microbiomes are everywhere. We talk about the human microbiome, right? We carry around 2 to 6 pounds of microbes. The cells of bacteria and fungi that are on us and in us are an order of 1 or 2 magnitudes larger than the cells that we have in our body. So microbiomes are the collection, the collective of all microbes, eukaryotes, prokaryotes, but also viruses are considered the microbiome. And not just the human microbiome is interesting because it affects our health. Microbiomes are everywhere. Microbiomes, I can't advance my slide. Now I can. Microbiomes are everywhere. We can find them in the soils, right outside in your garden, in your backyard. They're every inch of soil harbors order of magnitude of 1 million to more bacterial cells. We don't even know the amount of viruses precisely. Every ocean, every lake, every river has its own microbiome. And there is a microbiome also considered to be in the atmosphere. And I would argue that we haven't studied these collectives of microorganisms at all, and there's lots to discover. This is a very old quote from Leonardo da Vinci saying, "We know more about the movement of celestial bodies than we know about the soil underfoot." And I think that's true. We will have examples later where very interesting microbes and chemistry has been found right outside in the garden or next to a school. There's lots of things to discover. Okay, so in my lab, we are interested in these microbes and their chemistry and how we can find new antibiotics, new anti-cancer drugs, and new antivirals. Right now, obviously, we are in a viral pandemic, so finding molecules that are uniquely targeting a virus and not harming the patient is a big effort. So we are looking into microorganisms, and we brought to Whitney a collection of 5,000 unique strains of microbes isolated from the marine and soil environments. We are interested in looking for fungi and bacteria. So I introduced to you penicillin and streptomycin, so very potent antibiotics. These are the main player. And many of them are marine, and obviously being at Whitney, we want to expand our marine microbe collection. And aren't they pretty? They come in all shapes and all colors. And you have to believe me, they also smell interesting. So if you could smell this, there's a really nice aromatic version smell that is extruded by these bacteria. And all of them are living here at Whitney. So how do we process this? How do we move forward from these strains? So we start off with microbial isolation from the environment, from the oceans or from soil, and then we ferment these strains. So we have this large shaker here that's very similar to maybe beer brewing, where you give nutrients to the fungi and bacteria and you can grow them on large scale. And the beauty is with microbes, you can ferment them in really large scales. It's almost a sustainable culturing, because we can always go back and culture more instead of plants where you might go back and harvest the whole forest once you found a good drug. We then go in the laboratory. We are a chemistry lab, and we are excited to figure out which of those chemical entities is in our microbial soup. So we have lots of steps here where we separate. We have up to 100 to 200 metabolites, different chemical entities in those extracts, and we have to separate them. And then we do in-house a lot of questions about what are we having here? Is this a new antibiotic? Is this a potential cytotoxin for cancer treatments? Or do we have an antiviral lead? So early testing done in-house here at Whitney tells us, do we have a new drug lead? From that's the hard part for the chemist. So most of my students are either in the chemistry or pharmaceutical programs. We need to purify from these 100 metabolites, the one that's active, or maybe the 2 that are active. And they're different analytical chemistry different instrumentations we are using to figure out what is the molecular weight of a structure and what is its three-dimensional structure, so we can in the end propose a new drug lead and maybe pitch this for further development. So today I want to take you on a little journey to see where the microbes coming from that we are looking at momentarily. We will start with two projects where in the early stage where we are looking at microbes early, and later on you will see concrete drug leads that came out of these efforts. So first we start off with the Pacific Ocean and microorganisms that are associated with fishes. So there were big interest in the news in 2019 when we started a project that looks in what microbes are living in fishes and their slime and their mucus, and can that lead to new antibiotics. That was picked up by The Guardian and some other news outlets. But the background is very serious. We want to see the microbiome that is harbored with those fishes. Can we check out if there is any potential to develop a drug? This is done by graduate student Paige Manlair and undergraduate student Molly Austin. So it was a little bit new for us to look into fish. Normally we look for soil, maybe plants, and find out what bacteria are living there. But in this case, we truly had to process a lot of little fish. So as you can see here, our collaborator shipped us various fish, and we were isolating bacteria and fungi from them. So in this project, we are working in collaboration with Misty Page Tran. She is a professor at Cal State Fullerton. We also work with Jim Strother, who is a fish biologist, expert in that. And so we were interested in juvenile fish, shown here. And these juvenile fish were caught by Misty and her students, and they were swapped. You can see here, little cube pick, a little stick that is swapping these fish, and we were isolating a whole bunch of bacteria. So in total, I think 85 different bacteria were isolated, and we can identify what species there are. So we have from Pseudomonas to Streptomyces, very different microbes associated with these fish. They come in all different colors, as you can see down here as well. So we can grow these, and you can see here our culture vessels. We grow them, and these are three very colorful bacteria that we looked at. And what you can see on this chart is how these extracts are working against a cancer cell line. So we picked a colon cancer, a colon carcinoma cell line here, and you can see if we have no chemistry present, then we have 100% cell growth. The cancer cell is surviving. If we take extracts from these three strains, only one here in green is inhibiting the cancer cell line, and this is doing it quite strongly. So Molly Austin was a really good student. She looked into this strain a little more. So this is a Pseudomonas aeruginosa that was isolated from this pink surf perch, a coastal fish from the Pacific coast. And Molly could isolate these different chemical entities, with the first one being the cytotoxin. So this molecule here is the reason we are killing a colon cancer cell line. She also found several of these phenazines. These molecules are antibiotics. So from one story, from one strain, from one fish, we found both antibiotics as well as cytotoxins. So this is still an early stage. We are having 85 strains isolated, and over 40% of these strains are either antimicrobial or active against cancer cell lines. And so this is active research where we are trying to find new chemical entities. The next stop in our journey around the globe is bringing us to the Arctic Ocean. So we're going far north. This is a collaboration with the University of Norway at Tromsø. And here is a map of Europe, and the University of Tromsø is up here. This is the northernmost university there is on this globe, so the most far north. We work with a team of researchers, and they're going on different cruises and 2 cruises we were part of in 2016 and 17. Here is actually the team of collaborators that went on this cruise. And so they are driving or having a boat taking them out all the way here up north in the Arctic Ocean. So they're going to Svalbard up here. This is a better map that shows the expansion, and there are different collection sites on their cruise where they were sampling water and mud forests. So how do you do this? Well, you have either a little bit remotely operated vehicle, little U-boat here, or you have grappers, different mud grappers, or little clams like that. We can get cores drilled from the ocean bottom. And you can also see there are glass bottles, plastic bottles here on the side. These can collect water samples at different depths. And so from this, we also received various sediments and water samples. And then Zoe and Annabelle, at the time undergrad in the lab, were isolating all these different bacteria. Again, this is early. We just did this, it takes a few years to process everything. So we have 83 strains. And from them, there are a little fewer bioactive, but we are actively pursuing a few of them to find out the chemistry. So the Arctic Ocean is definitely a place that harbors microorganisms. So the last 2 stories, the last 2 stops on our globe travel are bringing us to concrete drug leads, to complete new molecules that might be new drugs in the future. So the first stop is another nice place to visit, the Mediterranean, right here in the Mediterranean Sea. We are visiting Gozo Island. So Gozo Island is here, south of the Italian Shoe in the Mediterranean Sea. And in that trip in 2007, a long time ago, we were able to isolate fungi that live within seagrass and seaweeds. So these algae here were harvested and we were able to isolate fungi. From these fungi, we see here on agar plates, 2 different ones were isolated from one single algae. And we were surprised to see that they produce chemistry. This is acrotoxin. This is a molecule that is known, chlorinated from the salt in the ocean, so it can incorporate chlorine. This is a known molecule. And the other fungus from the same algae produces these anthraquinones that do not have biological activity. So at the time, Paige was interested into combining these 2 fungi. They came from one algae. Well, what if we bring them back together? And that's what you see here on the agar plate on top. On the right is the yellow fungus I showed you, and on the left is this white fungus. And they do seem to not be able or wanting to touch each other. Here's a chromatogram that shows us different chemical entities. So we have a wavelength and we can absorb at different times. We can see different molecules. And Paige was able to see the anthraquinone, the compounds from the yellow fungus in the extract, and she could see new molecules that only were made in co-culture, combining both of these fungi. Since these were new molecules, Paige were able to name them, and she named them allianthrones after the fungus, Aspergillus alisius. And it was quite exciting to see we get over 35 milligrams per liter from this cultivation. So lots of chemical entity. The other 2 peaks here are related molecules. They're only subtly different in their 3-dimensional structure between those 2 halves. So why are we interested in these new molecules? These are, here's a crystal structure. So Paige was able to show the 3-dimensional structure shown here. This is by X-ray crystallography. So we can see the electron density in this molecule and could prove, yes, indeed, this is a novel chemical molecule that we were able to name. So we are interested in these compounds because they are potent cytotoxins. And Elizabeth Kaviser in my lab, she's running our cancer program. And so these new molecules were tested against 5 cancer cell lines. We're using a colon cancer, melanoma, lung cancer, breast and prostate cancer as a panel. And you can see one of the molecules, allianthrone A, is rather potent against the colon cancer cell line. Paige was also able to brominate these molecules. So here is our drug lead, our cancer lead, and she can make add with bromine, sodium bromide to the medium, make these molecules brominated as shown here. And that is neat. We can now compare these different chemical entities in their potential to be new drugs. We have done this in collaboration with the National Cancer Institute. And this is a slide that has a lot of information. I walk you through them. So what you can see here are 60 different cell lines that the National Cancer Institute is using to survey and assess compound activity and selectivity. So here are very different cancer cell types. On the center, you see a line, and every time our bar goes to the right, we are having lethality. We are killing the cell line rather selective. If the bar goes to the left, we are inhibiting the cell growth, but we're not killing the cell line. So now we have a very nice example for chlorinated and this brominated compound that Paige discovered. And we can ask, where are the differences? And it seems that the brominated compound here is more active in some cell lines, especially the renal cancer cell line down here. But to our surprise, or what's really highlight in the study, was that we are active against a triple negative breast cancer. So down here are the breast cancer cell lines, and the triple negative breast cancer cell lines, very aggressive, very hard to treat in the clinic. And we have very good response with these two compounds. And again, we can make a lot of these compounds, and we're working with April Reisinger at UT Health San Antonio. She is bringing these molecules into the mouse model to evaluate if these are new drugs to treat breast cancer. So this is a story from the drugs from the Mediterranean. In my last stop on our world tour, we're going back to my home, back to Germany. And there is a drug that we have been pushing forward quite far into development. So we are going to Germany, and we are looking at this molecule here, which we called Mensacarcin. This is coming from a bacterium. Here's an agar plate and the culture. This molecule was found in the early 2000s, and it's named Mensa after the cafeteria. So you can see on this picture here in the background is the cafeteria of the University of Göttingen, where I did my PhD studies. And this bacterium was isolated from soil right next to the cafeteria. So you don't have to go far, actually. It could be just right outside your building where a new bacterium, a new compound can be found. We were interested in Mensacarcin because it is a cytotoxin as well. So it's potentially inhibiting cancer cell lines. And here are shown a liver cell line, a breast cancer cell line, and a stomach cell line. And Mensacarcin is a new compound we found, and it was named after the cafeteria. And down here are two clinically used compounds in cancer treatments, and both of them are very not active, for example, against stomach cancer, while our compound is active. So we did a similar study going for the NCI assessment, and what you see here is again 60 different cell lines that were tested, and our compound Mensacarcin is very selective over here in the melanoma panel. So melanomas that were tested are all reaching a lethal concentration in our assay. The breast cancer models, not so much. So we are selective for melanoma over other breast or other cancer types. Melanoma is obviously important in Florida. It's aggressive skin cancer. Florida has the second highest incidence rate in the US, just following California. And one thing that might not be as much known is that every third metastatic brain cancer is actually originally a melanoma. So it is rather highly metastatic in the body. So we were interested in developing our compound further. And here you see some of the work Elizabeth did in cell culture. So we can see here, these are nuclei of cancer cells. And if they're treated with our compound, they're undergoing apoptosis, programmed cell death. And that's a good thing for a cancer cell. We clearly can see this here. We run some other assays showing that we metabolically impair the cells. Here are our melanoma cell lines. And at 1 micromolar treatment with this drug, the metabolic rate is completely slowed down. And we even break open the cells. Here we see the release of lactate dehydrogenase. So cells are disrupted with our compound and they are dying. So that's a good thing. We also are chemists, so we put a light bulb, shown here in red, on our molecule. So a chemical entity shown here is fluorescence, and we could couple that to our drug lead, to our Mensacarcin drug lead. And we still have down here shown the same cytotoxic abilities. We are still inhibiting cell viability, and we are still cytotoxic, even though we are a much larger molecule. With that, we can image these cancer cells, and here you see cancer cells under the microscope. We can label different parts of the nuclei. For example, in blue are the nuclear stain, in green we have mitochondria stain, and with our molecule and the red light bulb, we can see that they are overlapping, localizing in the mitochondria. So our molecule Mensacarcin affects cancer cells by going into mitochondria and affecting cell metabolism. So my last 2 slides, I know I'm running quite long. I want to show you, we were interested in stopping migration, stopping metastasis of melanoma cells. And what you see here is a collaboration with Virginia Tech, Dave Brown's team. You can see here cells, how they are marching, how they are migrating on little fibers. And so our collaborators have these tight ropes where cells are marching along. And we were interested, are cells able to move if we treat them with Mensacarcin? And you can see here 2 videos. I start them both. On the top, what you see are the cells sliding. So here are mouse muscle cells moving along these fibers just normally, just nicely. And down here, if we have the cells treated with Mensacarcin, these cells cannot migrate anymore. These cannot metastasize. And so that was really promising results. We just published this year, 2020, and we're thinking that this is a new migration inhibitor that will affect the cell metabolism, the glycolysis that's happening in our cells. And Elizabeth just recently could show that this is true. We have in the previous study, in the cell migration study, we have this mouse myoblast, and our molecule stops the uptake of glucose into the cell, so stopping the energy supply. And in melanoma cells, the ones that we are most interested in, the same results. Mensacarcin is stopping glucose uptake from the cell, from the cancer cells. So we are really excited that we have results like that. In my last slide, I have to apologize if you hadn't had a dinner. Melanoma is a very serious disease. And as you can see here, the current treatment of melanoma is Zelboraf, is this chemical entity here. The problem with Zelboraf is that a patient that has these really brutal lesions and takes the drug can be cleared from these tumors rather quickly, but then the cells become resistant. So resistance to this drug Zelboraf is really highly observed, and that is not a good prognosis for the patient. And what we found is, and Elizabeth did this work, is these are 3 different cell lines that are resistant to this drug Zelboraf, but our molecule, the Mensacarcin molecule, is still able to stop the cells from dividing, and it's stopping cell survival in a rather good dose. So we are hoping to bring this forward into mice model to explore Mensacarcin's potential as a new cancer drug. With that, I took you on a little tour. In the beginning, I showed you microbes that were just collected and they're waiting for discovery. In the end, we talked about concrete chemical molecules that might become new drugs in the future. And obviously, we are down here. We want to add more microbes from Florida, from the environment here. And so we're actively sampling And we cannot wait for this pandemic to be over and we can go out and have volunteers and students working on these projects. With that, I have to give a lot of thanks to my team here at Whitney. On top is shown the current members here at the lab, and I also show an older picture from students that worked on some of these projects before. I'm very indebted by our funding agencies, National Science Foundation and the German Research Foundation have sponsored us, and we are really grateful for that. And I'm also grateful for your time today, and hopefully you have some questions for me.
Super, Dr. Loesgen, thank you so much on taking us that journey and pointing out all the different microbes and funguses that we can see. So thank you for that. One question that's come up is, what is the total time that from the time that you collect the materials to the time that ends up in your laboratory, to even one step further to where you think that that compound may do something? What does that timeline look like?
So that's a good question. And I think in this slide, that was the first time I honestly say that it takes a long time. So some of the samples were collected 2015, 2016. It takes us several years to identify these microbes, to culture them, to have them in our collection, and then to screen them, right? Every of these assays is done by hand, by a student or postdoc or staff in the lab. So it takes 3 to 5 years from the sample we get to the microbe and then to some kind of chemistry idea. The next steps obviously take longer to test in vitro and hopefully in vivo. And this is usually a process that can take up to 10, 20 years easily to do this fundamental research properly in the beginning to bring a new molecule on the way.
Wow, so your work doesn't move quick then, does it?
No, unfortunately not. So it takes a while to study. To find new chemicals is hard. Many things have been seen before. And then to find something that is better than penicillin and better than the current drugs we're having is a challenge. But there is potential. And I think Mensacarcin we can push a little further.
Right. Wow. Well, as a follow-up question, so I think we can all agree that it's great to have these new drugs in the pipeline to treat a variety of cancers and other things. But how do we know that they don't destroy other cell types? So what is the additional steps and safety that's taken into place?
That's a good question, absolutely. So we usually test against a variety of other cell lines. Blood cells are really common to test, white blood cells, red blood cells. You don't want to harm these. And the cancer cells that we are having are rather aggressive, fast dividing cells. You can test for other liver cells and other primary cell lines to be sure that these are not affected by your drug. But in the end, it has to go at one point in a mouse model, and the National Cancer Institute has mice model for human cancers or for mice cancers, and we can test then how our molecule is shrinking a tumor or is affecting the health of the mice. If they are too harsh treated by the compound, these mice has to be sacrificed. There are very strict protocols and testing.
Wow, super. Thank you. I have an additional question. As I was fascinated by the photo that you showed about allianthrone with the symbiotic relationship of the two funguses and that clear delineation between the two. Are there in nature other symbiotic funguses that live together in order to both survive?
That is a very hard question. We see a lot of interactions in the microbiome. There's a lot of symbioses, but also everything from, being good friends and symbiotic, being good neighbors, to become pathogenic and being bad neighbors, harming the others. So there are beautiful studies, especially also in the ocean, about interactions, microbes, fungi, microbes, algae, where there is a happy together exchange maybe of nutrients, exchange of chemicals, but at one point it could break and then it becomes pathogen and a more sad interaction. From fungi, we know there are a lot of fungi. There are, we think we might have studied maybe 20% of the fungal kingdom. There's so much to do. And I wouldn't be surprised if there are fungal interaction. There are fungal parasites that parasitize fungi. So everything is possible.
Right. Wow. Wow. Great explanation. Well, since that one was challenging, then we'll go a little simpler. What made you actually interested in chemistry?
That's a good one. I was a pre-med, and that's interesting because when I teach, at Oregon State or here in Florida, you meet as undergrads a lot of pre-med students that have this goal to cure disease, to help people. And I did a volunteering internship for half a year in a hospital in Germany, and it was eye-opening for me. It was clearly that I was not able to be the person, to be the doctor in that room or the nurse in that room, for that matter of fact. And so I realized that I was actually more interested in what are these drugs? What are these cancer patients given? And why is it, killing selectively a tumor? And I fell back to my love for science. And chemistry was something that I was fascinated with. And it came back naturally to look for all the drugs that we are having come from pharmaceutical industry, come from developments out of nature. And so to look what's new there, what's more there, and to study these structures. The one I mentioned from Paige, it was a new structure. Nobody's ever seen it before. And to put this puzzle together and then to present it to the world as a new chemical was quite a thrill. It's really fun.
Yeah, great, great. We did have a question from the chat that's asking us, how long has Whitney been doing this kind of research?
Well, so, we moved here in September, and I think we bring in a big push in microbial chemistry. And there are other marine stations around us, actually. There's Harbor Branch and others, the Smithsonian Station, that have chemists on site. And obviously there is biochemistry has been here and geochemistry. But I think the natural product part is maybe new to Whitney with us moving here. And it's definitely an area we can expand more and engage the community more.
Super, super. Thank you so much, Dr. Loesgen, for your time tonight. I greatly appreciate it. So with that, I'll turn it over to Jessica.
Yes, thank you so much for your talk this evening. It was really interesting. And I think we all learned a lot, and we're going to get, I'm sure, more questions via email like we do after our talks. We did record this, so if you would like to share this with others, we usually post it in a few days on our Facebook and on our website. So if you know folks you want to share this with, we'll have the link up for you. I hope you can join us in January for our next Sip in Science, and we do hope that you have a happy and safe holiday, and thanks so much for joining us this evening. Have a nice evening.